In this lecture, we're going to talk about the liver, the gallbladder, and the pancreas, and a little bit about metabolism. So let's start with the pancreas. We've already talked about these cells, these lightly staining cells, which are the islets of Langerhans, cells that make insulin and glucagon to help regulate blood glucose levels. The pancreas is part of the endocrine system, but most of it, the pancreas, is made up of these darker staining glands, the acidic glands that make digestive enzymes that are secreted into the duodenum through the pancreatic duct. So in general, your pancreas is primarily epithelial tissue made of both endocrine and exocrine glands. So here we see the pancreatic duct that would carry those enzymes, sometimes called digestive juices, into the duodenum. So these digestive enzymes are helping to break down carbohydrate fragments that reach this region, also peptides and fatty acids. And we can see a close-up here of where the pancreatic duct is coming in, and it's before it enters the duodenum, it joins with the common bile duct carrying bile, and together they enter the small intestines. Okay, and we'll talk about the bile shortly. So that's the pancreas. The liver is also primarily epithelial tissue, and it is arranged into these structural and functional units called liver lobules. So you do need to be able to describe a liver lobule. It's shaped like a hexagon, so it's a six-sided structure. These cells are called hepatocytes. These are epithelial cells. And they line capillaries called sinusoids. If you recall from when we studied blood vessels, sinusoids are very leaky capillaries. And those sinusoids enter, the blood enters them from out here on the periphery. There are these what are called portal triads. Triad because there are three structures there. And we can see that better in this picture. Here we see the portal triad, which contains two types of blood vessels. A branch of the hepatic portal vein, which is carrying blood from the digestive tract, and also a branch of the hepatic artery off the aorta, the celiac trunk, and this is carrying oxygen-rich blood into the liver. And notice, this is very unusual, that the two are going to converge and enter the sinusoids here. So you have venous blood and arterial blood mixing. So this is this is part of a portal system, the hepatic portal system. So you want to bring this blood from the digestive tract to your liver in order to help process it, to remove excess nutrients, but also to help clean it up, because we don't just absorb nutrients in our digestive tract. We also absorb some pesticides or pollutants that could be in the food or water, contaminants, heavy metals. So these also get absorbed and are going to be, the liver helps in detoxification of the blood coming from your digestive tract. Now, it's not as if these hepatocytes can completely remove those substances sometimes, but often what they're able to do is to modify them so that later when that blood goes to your kidneys can remove it. So your liver and your kidneys really work together to help remove contaminants from the digestive tract. Okay, so the blood moves through those sinusoids, and then when it gets to the central vein, which is going to go into your inferior vena cava, it's been processed by the liver. The liver also makes bile. So the third part of the portal triad here is a branch of the bile duct. So bile is used to break up larger fat globules into smaller ones so that your lipase enzymes can get at them. So bile is for emulsification. It also contains some old heme, so it's which is going to enter your digestive tract and then be excreted with your feces. So it's also a way of getting rid of some old heme. So make sure you can describe the portal triad. What are the three vessels or duct that are part of it? Okay, this is rather blurry, I apologize, but I think you can see the main thing I'm trying to show here is the hepatic portal vein. So I wanted to remind you about that from the circulatory system. So if you recall, the hepatic portal vein takes blood into the liver, into your sinusoids, where it's going to be processed, and then it's going to leave your liver through the hepatic vein. So if you look at the hepatic portal vein and look at, if you follow it back, notice how many branches feed into it, coming from different parts of your digestive tract. You see the stomach here, the large intestines, the small intestines as well. The blood all feeds into that hepatic portal vein. This is what the liver looks like under the microscope. So here we see the hepatocytes in pink, the sinusoids where the blood will course through into the central vein. Okay, so the liver, as we mentioned, detoxifies blood coming from the digestive tract. That is the function of the hepatic portal system, where the first capillary bed is in your digestive tract for absorption. The second capillary bed are the sinusoids in your liver for helping to process that blood. And as we see in this slide, there are many other functions of the liver, many of which we really don't go into in much detail. But the liver does help process sugars, lipids, proteins, detoxification of drugs, hormones, and things absorbed through the digestive tract, the excretion of bilirubin I mentioned in bile salts for emulsification of fats. Our liver does have glycogen in it, enough for 5 to 6 hours of glucose, because remember, glycogen is the storage form of glucose in animals. So you see that it has multiple functions. Okay, so that was the detail which we're going to go into the liver. And the gallbladder, before I forget to mention it, is pretty simple in its function. It stores bile. It does not make it. It stores bile made in the liver, and then when you need it, excess bile, you can release it into the cystic duct and down into the common bile duct. Okay, so the other main topic I wanted to talk about in this lecture is some basic metabolism. So this slide shows the 6 main types of nutrients that the body needs. Of course, water, the basic universal solvent of life, and also a participant in many chemical reactions, like hydrolysis, using water to, for example, in the breaking down of proteins into amino acids. Sugars, used primarily for energy, but they can have other functions, or carbohydrates. Lipids, for example, a major component of cell walls. Also, triglycerides store a lot of energy. Proteins, which have multiple diverse important functions, from structural to regulatory, like enzymes and hormones. Minerals, sodium and potassium, needed for things like nerve impulse conduction. Calcium, which has, for example, an important role in muscle regulation of contraction in muscle, or in making the composition of bone hard. Various vitamins, which we'll touch on briefly. These are the 6 considered main types of nutrients. Review of some vocabulary that you probably learned in basic biology. Metabolism is just the sum of all the chemical reactions that go on inside the body, and it's broken down into 2 main categories. Anabolic, where you're taking smaller components and combining them into larger molecules. For example, protein synthesis, connecting amino acids together to make proteins. That's anabolic, and it requires energy in the form of ATP. Catabolic, on the other hand, reactions are breaking down larger organic molecules into smaller ones, like proteins into amino acids, and that releases energy. So, for example, breaking down glucose to carbon dioxide and water releases energy used to make ATP. Okay, so the body uses the energy released from catabolism to drive anabolic reactions. And our cells are fairly resourceful or efficient in that they can extract about 40% of the energy stored in, for example, a glucose molecule, stored in the form of ATP, the universal energy molecule for cells, and then the other 60% is in the form of heat. So again, ATP is really what the digestive system is all about, obtaining, or to a large extent, obtaining nutrients that can then be used to make ATP by catabolic reactions, and then that ATP can be used to make molecules that the cells need, like proteins. Okay, a review of cellular respiration. We already reviewed this when we covered the respiratory system, so but do make sure you know this basic overall equation of cellular respiration. So the carbohydrates that you consume can be broken down or catabolized by your cells to make ATP, or you can store them. For example, your liver can store them as glycogen, or you can convert them to fats or to amino acids. This is the overall pathway of cellular respiration. So we are not going to be reviewing this, but it is something you should know. The different stages of cellular respiration: glycolysis, Krebs cycle, electron transport chain. Okay, just some definitions really here. Glycogen, again, that's how we store excess glucose, or at least some of the excess glucose. You can also convert it to fat. And some organs are able to actually make glucose from other organic molecules, like some of the other, some amino acids, for instance, and that's called gluconeogenesis. Neogenesis, the formation of glucose from other organic molecules. So your liver and your skeletal muscle cells can do that. It's important that some of your organs be able to do that, because glucose is such an important nutrient, especially for some of your body cells, like your cardiac muscle cells and your neurons. Triglycerides, another one of the major nutrient types. This shows a triglyceride, which recall is made of glycerol, 3-carbon glycerol, and then 3 long carbon-hydrogen chains, the fatty acid chains. So these are good for storing energy, but you can also take off one of these fatty acid groups and replace it with the phosphate group and produce phospholipids, that is the main backbone of your cell membranes. If the covalent bonds between in the fatty acid chain does not have any double bonds, we say that it's a saturated fat. If it does have one or more double bonds, it's referred to as an unsaturated fat. They do have different properties. Unsaturated fats tend to be liquid at room temperature, saturated fats solid. Unsaturated fats are thought to be in general healthier for us. We consume both in our diet. Cholesterol is also a type of lipid. I do want you to know that while important in the body, cholesterol, it is something we need. We do not use it for making ATP. So you do not break cholesterol down like you do fatty acids to make ATP. You do, however, use it in making your cell walls and in making steroid hormones like testosterone and estrogen. It's also used in making bile salts for the emulsification of lipids. So it is important, and it's in general a concern for many people who have high cholesterol levels. But it's important to remember that high cholesterol levels are not good for us, but cholesterol itself is an important molecule that we need to be healthy. So let's talk a little bit more about cholesterol, since it is something it's commonly measured in assessing health. So lipoproteins are a combination of, just like the name suggests, lipids and proteins together. And the main reason we form complexes between lipids and proteins is so that we can transport them in our blood, because your blood is mostly water and lipids are hydrophobic, so they're difficult to transport on their own. But if you complex them with a protein, then they'll be more soluble in your blood. Okay, and there's different kinds of lipoprotein complexes. So you do need to know the difference between them. Chylomicrons are complexes carried by our lymph. They're picked up in our lymph capillaries, and they transport lipids that you absorb through your digestive tract to your adipose cells. So those are called chylomicrons. Okay, and remember, lymph does eventually go into your blood. VLDLs are another type of lipoprotein complex. Their name does not really tell you much about their function. It really has to do basically with how heavy they are. VLDL stands for very low density lipoproteins. So they're lighter than the two below that we'll talk about in a moment. But that's what they're called, VLDLs. And they transport triglycerides to adipose tissue. The proteins that make up the VLDLs are primarily made in your liver. And so you're going to complex triglycerides with that, send them out from your liver, and your adipose cells are going to take up some of those lipids from the VLDLs. But not all of the lipids. And actually what's left, triglycerides and also cholesterol, is now what we call LDL, low density lipoproteins. So it's a little heavier than the VLDLs because some of the lipids are removed. And this is the form in which we transport a majority of cholesterol to our cells. Okay, so they need it, again, for making cell membranes, for instance. So your general body cells have LDL receptors on their surface that the LDLs will bind with, and then from there they take up the cholesterol. Okay, now we'll talk about when these levels are too high in a moment, but LDLs in and of themselves are they have a useful function, transporting cholesterol to cells. And then HDLs, high-density lipoproteins, their job is that they're made up of like a protein sac that is sent out from the liver to pick up cholesterol and bring it back to the liver to be used to make bile, which is then going to be eventually eliminated. So HDLs contain cholesterol that is going to be removed pretty shortly from the body. So when you have your cholesterol levels measured, they don't just look at your total cholesterol level. They're also going to look at your LDL levels and your HDL levels. Because if your LDL levels are higher than normal, then that is considered a risk factor, because too many LDLs hanging around in your blood vessels, not needed by your cells, not being taken up, or their cholesterol not being taken up and used by your cells, will be in the lumen of your blood vessels, and the lipids can get deposited on your blood vessel walls or in your blood vessel walls and form plaque and lead to things like atherosclerosis, or then that could get calcified and form arterial sclerosis, hardening of the arteries. So high LDL levels are considered bad. High HDL levels, on the other hand, are considered good because the cholesterol they contain is heading to your liver to then be excreted with bile. Okay, so those are the lipids. For the amino acids, which we need for making proteins, we're not going to go into that much, but I do want you to know these two terms, essential and non-essential. They're a little bit misleading in that it makes it sound like the non-essential amino acids are not really needed, but what they really mean is whether or not you can make those amino acids yourself from precursors. If you can, then they're considered non-essential because you can make them. But if you can only get them through your diet, then they're considered essential amino acids. Okay, so complete proteins contain all the essential amino acids. Incomplete do not. Okay, so those are the organic nutrients, and then just a little bit about vitamins. So this is very minimal. If you take nutrition, you'll learn a lot more about these. But in our course, I just want to go over a few things about vitamins. First of all, they can be divided into the water-soluble and the fat-soluble group. The water-soluble vitamins are the C and B vitamins. Those you don't have to worry as much about in terms of dosage, because you can pretty easily remove the excess of these through your urine. Fat-soluble vitamins, on the other hand, you have to be careful of because they're not as readily eliminated from the body. And so if you have too much of vitamins A, D, E, or K, that can lead to problems and be unhealthy. So know your water-soluble from your fat-soluble vitamins. And then for each vitamin, I may ask you to give one function. So you can use any function that you read about in your book or that you know of. But I'm just going to go through and give you an example, but you don't have to necessarily use the one I give. So vitamin C is primarily used as an antioxidant to help get rid of what they call free radicals. These are byproducts of all the chemical reactions, the metabolic reactions going on inside cells, and some of them are can have negative effects on your cell, so we need to get rid of them, and so vitamin C helps do that. It's called an antioxidant. The B vitamins, and notice it's plural here because there's many subtypes which we are not going to go into in here, like riboflavin and niacin and pantothenic acid, and there's many subtypes for vitamin B. But in general, I'm going to lump them together and say, in general, one function of many of them anyway is to serve as coenzymes. They are needed by certain enzymes, especially they're important in cellular respiration, where they help. They're parts of important vitamins. They help carry high energy electrons, so they serve as coenzymes. And then for the fat soluble vitamins, vitamin E is, like vitamin C, an antioxidant, but it's found in fat soluble regions, so like in membranes. And so C and E are related in that they have these antioxidative functions. Vitamin A is needed to make visual pigments in your eye. That's one function, major function. Vitamin D is very important in helping to properly mineralize your bones, to move the proper amount of calcium into your bones. And vitamin K is a vitamin made by some of the bacteria that make up our microbiome, and it has a role in blood clotting. So vitamin K is needed for hemostasis, for some of the steps in the blood clotting process. So as long as you know one function of each vitamin, you'll be fine for the test. Okay, so that completes our very brief overview of metabolism.